The first step of cellular aerobic respiration is glycolysis. To start, we will need 1 molecule of glucose. A molecule of ATP comes in and splits the sugar into 2 3-carbohydrate long chain bases with 1 phosphate on each end. Another phosphate from our diet is added to the chain. Next, the chain is oxidized and loses 2 electrons. These electrons bind to NAD+ to form NADH. Molecules of ADP come by and receive molecules of phosphate, forming usable energy in the form of ATP. The conversion of ADP to ATP is done via substrate-level phosphorylation. This leaves us with a net gain of 2 ATP molecules and 2 pyruvate molecules that move on to the prep stage of the Krebs cycle. Each of our pyruvate molecules goes through the prep stage of the Krebs cycle. First, pyruvate dissociates into CO2 and acetic acid. Acetic acid oxidizes 2 electrons to form 2 more molecules of NADH. Coenzyme A comes in and binds to the acetic acid and forms acetyl coenzyme A. This moves on to the Krebs cycle. Coenzyme A drops off the acetate onto the Krebs cycle, where it will bind to oxaloacetate to form a 6-molecule long chain. The chain oxidizes 2 electrons to make 1 molecule of NADH and releases 1 molecule of CO2. The chain releases another molecule of CO2, as well as oxidizing electrons to make another molecule of NADH. At the next step, the chain drops a phosphate for the creation of 1 ATP. Next, the chain oxidizes 2 electrons to make FAD into FADH2. And finally, the chain reduces a molecule of NAD+ plus into NADH. Oxidative phosphorylation is where most of the ATP is made. The electron transport chain is the first part. All the electrons from NADH and FADH2 are collected and dropped off here. Since oxygen is highly electronegative, it pulls the electrons through the membrane proteins in order to put protons into the inner membrane space from the matrix of the mitochondrion. All of this potential energy is stored in the form of a concentration gradient. In chemiosmosis, the protons pass through ATP synthase, where the energy from the concentration gradient is built up and made into ATP. Fermentation is a form of anaerobic respiration. In lactic acid fermentation, NADH is oxidized and gives its electrons to pyruvate, which makes lactate. NADH can now accept more electrons from glycolysis. The main purpose of fermentation is to prevent the buildup of NADH. It is also very inefficient and only 2 ATP per glucose. In alcohol fermentation, pyruvate is split into CO2 and acetaldehyde. NADH is oxidized and gives its electrons to acetaldehyde. This makes ethanol and allows NADH to pick up more electrons.